Hello kids, today we are going to study about development of placenta. Now these are the competencies which we will cover. I am going to take this in two different lectures. For lecture 1, our objective is at the end of lecture every student should understand and answer development and functions of placenta. So now what is placenta? When we study on gross, we can see fetal surface. This is the fetal surface and this is the maternal surface. On the fetal surface, when we see at the periphery, we can see membranes. Okay, these are the part of the membrane is removed. You can see attachment of the umbilical cord and you can see the fetal blood vessels. So this is fetal surface. Then on the maternal surface, we can see number of these areas. These are called as cotyledons. Okay, these are cotyledons. So now what is placenta? Placenta is materno-fetal organ which starts to develop at the beginning at the implantation and it shades off with the fetus at the delivery. Okay, now this placenta measurements, it measures about 15 to 20 centimeter in diameter, weighs about 500 gram and its thickness is about 3 centimeter at the center. Okay, now placenta could be thin, it could be thick, that all depends, and various anomalies we can calculate from its excessive weight or excess diameter or thinness. Okay, so normal we should know. Then we already have studied about the fetal surface as well as maternal surface and the membranes. These maternal surface, they are called as cotyledons, 15 to 20 cotyledons, right? Now, development. In the development, see there is fetal aspect and maternal aspect. Okay, so there are two surfaces. On the fetal aspect, there is chorion frondosum. On the maternal part, it is called as decidua basalis. Details of this we'll study. So, at the time of implantation, the developing embryo is in the form of blastocyst. It is having peripherally situated trophoblastic cells and inner cell mass or the embryoblast. The maternal aspect is already in the phase of decidualization. That is, it is the uterus that is edematous, it is thickened, the endometrium is thickened and the stroma is edematous, vascular, the glands they are increased. So the uterus is ready to receive the developing embryo. The changes in the cell mass, this we have already studied. As the development of this placenta continues, at the same time the changes they are occurring in the cells of the inner cell mass in the form of formation of membranes, formation of amniotic and chorionic cavity, secondary yolk sac, bilamellar embryo. As the development proceeds in the third week of development, trilamellar embryo, connecting stalk. So these all changes they are occurring in the cells of the inner cell mass. Today we will focus mainly on the changes which are occurring in the trophoblast. So this is the inner cell mass which is fully developed by the end of second week. Okay, and we will focus ourselves here. Now as the development proceeds, there is formation of the extra embryonic mesoderm on the outer aspect within this trophoblast. Okay, this extra embryonic mesoderm, it further divides itself into somatotropic and the somatopluric and the splanchnopluric extra embryonic mesoderm with the development of extra embryonic coelom. Okay, now why this is important? Because this extra embryonic mesoderm, this we are going to consider or study during development of villi. Okay, so the changes which are seen in the endometrium. Okay, now endometrium we already have seen decidualization, maternal blood vessels they develop, and in the previous slide which we have seen, see this is the trophoblast which is developing and the spaces they appear in this trophoblast, and these spaces they are further these maternal blood vessels, they enter into these spaces. These spaces are called as lacunae. Okay, just remember this thing. So the changes which are seen in the endometrium is the maternal blood vessels, they open in the lacuna. What is lacuna? Lacuna is a space which will develop in the trophoblast.